Okay, well, good afternoon, everyone. Thanks for joining us for the first Wheatland seminar of the semester. Most of you know me, Tony Guay, remote sensing specialist in the Wheatland Geospatial Lab. And this afternoon, we're very fortunate to have Dr. Peter Nelson with us for this afternoon's seminar. Dr. Nelson is the Forest Ecology Director at the Spatial Institute at Acadia National Park. Peter oversees an assessment of the current health of forest in eastern U.S. parks from Maine to Virginia in partnership with inventory and monitoring networks of the National Park Service. He is analyzing 12 years of data on tree canopy and understory growth and biodiversity from more than 1,400 forest plots established in more than 30 national parks and historic sites. This assessment, expected to be completed this year, will inform a conceptual model to guide forest management decisions in the future across eastern parks and beyond. Peter previously served as associate professor at the University of Maine, Fort Kent, where he expanded a research program focused on new methods for quantifying forest resources using a wide variety of remote sensing approaches and platforms, from drones to fixed-wing aircraft and satellites. Peter earned a bachelor's degree from the University of Montana in 2004 and a PhD at Oregon State University in 2013. And with that, Peter, I'll turn it over to you.
Thanks, Tony. Thank you, everyone, for attending. And like many folks, it's been an interesting year, and we've been continuing to work. So I'm going to share my screen and show you some of that. Let's see here. Can everyone see my screen okay? All right, I'm going to go video free to save bandwidth. Okay. And if I am not coming through okay, I'm going to try to monitor the chat. It kind of goes invisible. So I'm going to try to keep tabs on it. So today I'd use the title Maine's Landscape of Light, mostly because of a paper Doodoo and I just were co-authors on, in which we use the term light to describe what we do. Here are some instruments measuring light over here that I'll talk about. Let me just get started. The structure of today's talk will basically start with the premise that there's information in light. Light tells us things about our environment that is useful, and that's basically fundamentally what I'm going to talk about today. Which part of the light are we studying, and what kinds of ways that are new to measure light am I going to talk about today? What kind of information can we derive from that light? And the work that we're doing to measure light to address the needs of Maine that might be within the kinds of new information we get from these sensors, and then what future directions might Maine think about for using these kinds of light measurements. So the goal really for all of these things is to understand the surface of the Earth across space and time, to really understand new things about it, like answering questions we haven't answered before. And I'm going to talk about focus questions with that kind of domain for Maine today. But the basic themes for the talk are that remote sensing of diverse targets is something, that is the main goal of this project. It isn't just one particular thing that these tools that I'm going to talk about are useful for. I'm going to talk about spatial and spectral resolution broadly. I don't want to get too much into the weeds of the technology, but just to get us thinking about how spatial and spectral resolution interact really are important concepts for people to understand, so that they can understand what these things can and can't do. Any given type of data from this type of imagery, really depends on the spectral and spatial resolution. And then I'll talk about some applications of these to remote sensing in diverse targets here in Maine. And the take-home message, of course, is that these things enable visualizing diverse targets that help us understand our environment and do things that are useful. The main thing I want to start with, though, is acknowledging some of the people that made this happen. These are current and former lab members, and in fact, I think all of them are former now. But Catherine Chan here was a pilot and analyst, did field work and writing. She's a PhD student now at University of Nebraska and an alumnus of the School of Forest Resources. Kayvon Smith is a pilot analyst in fieldwork. He has switched gears and is on a degree track towards a mental health degree. And he did a lot of work for us for several years. Jane Pettit ran the lab as a sort of lab manager and did fieldwork and analysis as well. David Parody was really the first student to really help get things off the ground, literally and figuratively, was the first pilot to go with us to Alaska and really try these systems in adverse conditions. And then Patrick McAuliffe really helped get the first initial instrument set up, which was the lab-based measurements. So I just want to acknowledge those students, all of whom are University of Maine, either graduates or were students here. I would also like to acknowledge the people who have funded this work, as well as places you could go to find more information about this, including the Schoodic Institute, NASA, the School of Forest Resources, University of Maine at Fort Kent, and others. So without further ado, what is light? Light is a particle and a wave, which, you know, obviously is not the domain of this talk, but it's important, especially the wave part, because as this picture denotes, energy is variable with respect to wavelength. So x-axis here is increasing wavelength and decreasing energy. Longer waves have lower energy. But thinking about waves in generally is what we need to start doing in this talk, but mostly as they tell us about what they bounce off of and look like to different measurements. And the part of the electromagnetic spectrum that we're going to talk about today is called the visible near infrared and shortwave infrared, from around 400 nanometers, which is this black line here, all the way over to about 2,500 nanometers, which is around here. So we're not using all light, we're just using the light between these wavelengths. And I'll talk more about how light is measured in a minute. But the way to think about them is in individual bands or colors. And this little blowout here is showing where the visible range of the electromagnetic spectrum is, where our eyes see. And the basic idea is that our sensors really just see the red, green, and blue, the ones that we have in our pockets, the cameras we have. And we're going to use sensors that see not only more colors, but out into parts we cannot see with our eyes. So different wavelengths, of course, tell us different things. And this whole idea is obviously taken to a much larger conclusion, where all parts of the electromagnetic spectrum are used by scientists to study things outside of just applications in the visible and near infrared. Stars are studied with gamma rays. Stars are also studied with radio waves. The Earth is studied with radio waves, and everything in between. So we're just really just taking a small slice of the electromagnetic spectrum and looking into it. So as I said, the visible and near infrared is the focus for today. So the thing about light and information is the following, which is that if there is information in light, do we get more information as we add more light, or do we get a diminishing return of information or an increasing amount of information with additional light? And the reason this is important is that it's really hard to measure all these other types of light with different expensive sensors. So these considerations are both scientifically and practically important. So if you have more light, do you have an increasing equal amount of increasing information you get out of the light, where this red line represents increasing amounts of information with more colors or bands? Or maybe as you increase light, every additional band really just adds tons more information. Or maybe just adding a little bit more of some additional electromagnetic spectrum information tells us just vanishing a little more than the last bit, and maybe it's not worth it. Of course, this is very simplistic, but I think it's important to start with what the considerations are with respect to how much of the electromagnetic spectrum you cover and how much you actually get out of it. Now, if you were to look at something with colors into the infrared, you would possibly see things that would look something like this. And now this isn't a picture, but it represents the brightness of light in those wavelengths we cannot see. So for reference, here's the visible range, and each squiggly line is one species of tree from Maine, just to show you an example. And the x-axis here is wavelength, and the y-axis here is reflectance. It goes from 0 to 1, 1 being absolute bright white surface. And you see these squiggly lines correspond to different surfaces, different plants, and places like in this familiar green region is obviously also slightly different. And the magnitude and the variation of these signatures really tell us a lot about the different surfaces and properties of those things. And so that's the starting place that our talk starts to take dimensions of these squiggly lines and look at them in pictures. Now, looking at the electromagnetic spectrum and thinking about which part of it is useful and what it tells us is only part of the question. We really need to also think about how those things are measured in space. And the way we usually get information in pictures anyway is in pixels. And of course, pixels are squares of different sizes, or at least they usually are. And without going into the abstraction of why that is, we could take a picture of somewhere that has various shrubs and of different sizes and low vegetation and some scraggly things that are, probably bushes and things. Which size pixel would we need to be able to resolve these different things? And of course, also know which parts of the electromagnetic spectrum would we need to be able to separate these things. And if we sort of think about them in terms of these 30-meter pixels, these bigger pixels, these blue squares, or 4-meter pixels in this white square, or 10-centimeter pixels with like this red square, and we overlay those on an image, we can clearly see that, smaller pixels are better, but at the same time, a bigger pixel will do for many things. And so the question is also not only which parts of the electromagnetic spectrum do we care about, but which size pixels. And without going into the physics, there's a lot of trade-off with those two things. And the same thing happens, oops, an N dropped here, but the same thing happens with pixels as it does with light. If we have higher resolution pixels up here, do we get tons more information? Or do we really not get much of a return on our investment? And so if smaller pixels, if we get bigger and bigger pixels, do we get, smaller and smaller pixels, do we get more and more information? Is there really a more and more return on our investment if we have smaller and smaller pixels? Or is there really a place at which there's no additional new information? And again, these are simplistic, but those two things interact and control what we're able to derive out of images. So if we combine the two concepts of hundreds of colors, each with its own perspective, patches of different sizes, each potentially with a different individual or thing that it represents, the spatial and spectral resolution complement one another, but they're also at odds with each other. And they're at odds with each other because of the way that cameras work. And we won't go into it, but we have to think about how those two interact practically. And again, these images showing here represent the same place in different wavelengths. And it's just the beginning of what the visual representation would be if a human could see with all these different wavelengths. And so just here are two images that we'd be probably more familiar with, like a red, green, and blue, and a color infrared. But these come from other parts of the electromagnetic spectrum in the visible and near infrared, and they tell us different things about those surfaces. And so if we were to combine different size pixels with different bands, we would see different things in these pictures. And that's what I wanted to make sure my audience knew before we departed into some of the details. So I'm gonna first preface by saying that I borrowed some slides from David Thompson at JPL, who is someone who does a great job in this tutorial. It's available on GitHub for open to the public, showing how some of the spectroscopy, which is what these kinds of cameras are called, works. And I don't want to use words like that because they're fairly complicated. So I just want to say that, the light coming into our atmosphere controls that first part of my talk, which is which part of the light are we going to use to measure stuff? And what I want to demonstrate here, and what David's slide demonstrates nicely, is that on the x-axis you have wavelength, on the y-axis you have the amount of energy, and this red squiggly line shows where is the energy that is available for doing stuff in the electromagnetic spectrum. And this is the energy coming from the sun, and most of it is available in the UV and the visible. And what is being emitted by the surface of the Earth is this green line, emission back into space. And these squiggly lines here show some of the reflectance properties and windows in which there's no energy for us to do work with and measure stuff. The point here is that most of the energy is coming in right here. And so we have a lot of potential to do work and understand things between 200 or 2000 nanometers and the UV. And that's really why we want to use those wavelengths, because there just is a lot more energy there for us to measure. The other question is, how wide should these colors or bands be? You know, knowing that we need to separate lots of different things that have lots of different colors, we would want lots and lots of different bands. But as I mentioned, it's very challenging to do that. And so if you want to compare two surfaces that you would measure using two cameras with just a few colors, like this figure here, versus a camera that had hundreds or thousands of colors like this signature here, you would be able to visually compare what you are gaining or losing. So what you're looking at here is two scans of one surface, a mineral mixture, which is an iron mixture. And the bands here represent the Landsat bands, which are obviously wide bands that are sprinkled through the same region we're interested in. And if you looked at iron-rich mineral, the same mineral with a sensor like the one I was showing you or talking about earlier, the signature would recover this nice bumpy line that would tell us a lot more about what that surface was. And so the little table down here at the bottom shows you that the trade-offs between which number of bands and colors you use is represented in a multi-band category, or what is now called imaging spectroscopy. And I'm going to use that word now and then, and other people often use the word hyperspectral, but in this talk, I'm just going to try to talk about lots more colors of light. And that's really what I'm talking about when I say that. So we're going to be talking mostly about the color regime here on the right, where we can see lots of different nuances in the signature of the surfaces that we're looking at. And we're going to refer to that kind of tool as either imaging spectroscopy or hyperspectral, but only when necessary. So how does this technology work? And assuming you're sort of on board with the idea that it's worth trying to measure hundreds of colors rather than just a few, it's important to note that the way the data are collected with a camera that measures these hundreds of colors is quite different than the way that a camera works that you have in your pocket. And the main difference is that it operates like a paintbrush, in which it's only measuring a very thin strip, almost like a paintbrush, and you have to move it one direction to paint along or collect pixels in this little black strip. And each pixel in this strip measures a whole bunch of colors, like this x-axis here, at different brightnesses, which is this y-axis here. And so the main idea is each pixel has one of these squiggly lines. And so we're going to talk about two different instruments here, the cameras and the instruments that just point at a single thing and make a squiggly line like this. But all of them do the same thing, which is that they measure hundreds of colors. Now, one of the things that we want to do, again, in a place that's big like Maine, is to be able to maintain the resolution of what we care about, like tiny little plants, but also over a large area. And for those of you who have seen my talks before, you've seen this figure in some form, which is borrowed from this great paper, which or adapted from it. And it shows that on the x-axis, you have spatial resolution. So these are smaller pixels and these are bigger pixels. And then you have how much of an area these images cover. And so if we want to do these things in that I'm about to talk about in Maine, we need to think about it in terms of a synergistic approach to scaling up images that we collect at the lowest level up to the bigger level. And so what you can see here is a picture showing that same template area and some examples, pixel sizes here superimposed on that vegetation from a satellite image. Here's that 30-meter pixel. Here's an aircraft pixel, that's this white box here. And then a UAV or unoccupied aerial something is a 10-centimeter pixel. And the idea here, again, is to make sure that people realize that you can't obviously fly over the whole state of Maine with a drone, but you also want to tell things about the whole area at as fine a scale as your application needs. So my point here is that we are working across these scales in the applications I'm about to tell you, because we knew from the get-go that no one of these scales was going to get us to where we wanted to go, even if we had all the different colors that we would need in a big pixel. We'd still need the small and medium sized. So here's a few pictures to show you. I already showed you once, but I'm going to show you in more detail what this experience of seeing in these other colors would maybe be like. So here's a regular picture of some low vegetation with some shrubs using regular channels, red, green, and blue, measured in nanometers. And here are the red, green, and blue channels. Now, if you switch to another familiar display, which many of us, especially those who have worked in forestry for some time, have looked at color infrared pictures. And there are many people who can collect images in the color infrared, and it's a very useful set of wavelengths, especially this 860 nanometer range out there where green vegetation is quite bright. And so you can see how different things look in those wavelengths. Now, when you add some other bands further out along the infrared, you get some really fun nuance. And these bands were selected based on some a priori knowledge of which parts of the electromagnetic spectrum would be useful in separating vegetation. But the point is that, if you were able to see in these colors, your experience of the environment would change. And here's one last, which is trying to contrast more photosynthetic organisms that are either vascular and non-vascular. And there's actually quite a few non-vascular plants in this picture, which are things like lichens and mosses in the understory. So now that you've gotten some basic idea of what the technologies are that we're going to use, we need to go over some very basic remote sensing terms before we can move on. So the idea of using remote sensing, whether it's the colors I've talked about or any others, is using the energy bouncing off a surface to measure something. It can measure it at different wavelengths, like we've said. We know that there's hundreds of colors. We've talked about that. So where could these diverse targets be detected by remote sensing? Anywhere, really, that you could see them up from above, as long as they're not being blocked by something. And that could be in the lab or in the field. So what kind of questions are we going to talk about today specifically that these kinds of remote sensing tools could be useful for? Potentially the ones that have already been shown to be working, which are plant health, plant identity, geology, water, and soil chemistry, things like that. But more specific applications that maybe jive with the scientific words that we would use to say, what would these images provide us in terms of applications? Hydrological buffering is a good example where we can understand where there might be hydrological blowout based on the vegetation. We can understand nutrient cycling. We can understand things that are in the ecosystem better by separating the species that are there. We can understand things like permafrost in the north and soil properties everywhere else. We can also do things like genotyping and chemical analysis and physiological measurements of living things with imagery, which that's hard to believe, especially the genotyping. But these are just some high-level things that these cameras are capable of doing. But we'll get into the details in some applications in Maine shortly. So this, for those of you who have been through this before, is the introduction of how we do these things. And it's a group of us, I would say, that I've referred to as the Laboratory of Ecological Spectroscopy, which is people, code, and hardware. And so the people I already introduced, the students, but there are many others who are faculty and collaborators across applications that I have not mentioned. There's instruments, which are important, but I don't want to spend a lot of time talking about them. But there's a drone-based imaging spectrometer, and there's a ground-based spectrometer that doesn't take a picture, but takes a scan. We've written software, but others on this call also have, and actually some of our software uses that software. And there's many different packages out there to do work with these data, but we've written some. And the things that we've done with these applications has been really to ask two main questions. What are the targets that are present, like species, and how much of a target is present? And really those two questions are just different slices through the same sort of high-level goals that we've talked about today. So how do we do these types of data collections with the instruments and applications I'm about to tell you about? Well, it works very simply by being able to go out into the field with the instruments. We scan stuff either on the ground and from the air. We make sure that we make measurements on the ground of what those things are that we scanned and flew over, such as where's this plant or where's that plant, or taking tissue samples or soil samples and making sure we know exactly where they were taken from, and we make chemical measurements of those. We process those data, both imagery and those reference data, and we analyze them in a model, and then we produce maps. And it isn't any more complicated than that. There's a lot of detail, but that's how our workflow with hundreds of colors towards an application that would be useful to Maine works. And we call that ecology. So just a little bit down into the weeds for those of you who care, there's a little bit of a workflow we worked up to make us a little bit of a, I wouldn't say a leader, but we've figured out a way to quickly take the scans from the instruments, and with a known plant and a collection of spectra, we can process it into a spectral library, do some cleaning, and do a bunch of derivatives that we plug into a relatively simple machine learning classifier, and then we point that at an image, and it provides us a pixel-based estimate of what's in each pixel. What we're working on right now is how to take those pixels and assign them into like objects, like this tree and that tree. I'm not going to talk about that today, but just a little bit about under the hood. A little more about the technology. For those of you who haven't seen this talk, this is an instrument, or this part of the talk, or about my lab, this is an instrument that we use to measure plants and leaves. It's called a spectroradiometer, and it has thousands of channels. And this, this is the wrong number of bands. It has 2,151 bands, but only 1 pixel, so there's no image. And these are the data that it makes. So these different kinds of surfaces, bare rock, this is quartz. You can see there's quite different signatures of different things, which is just for demonstration purposes for those of you who have never seen those data. The other instrument that we use is this one, which is an airborne instrument on a drone that has 324 colors over a narrower range of the electromagnetic spectrum. And we couple the data from the spectroradiometer of different targets with the imagery to make the maps that I showed in the models I described. And this little red box shows the part of the electromagnetic spectrum the camera can see. So we don't get to use all of the shortwave infrared, but the visible infrared is the main part of what ecology uses. So here's some examples in Maine. We're going to go over 4 examples: intertidal macroalgae using ecology in various degrees, ash health, which is a tree species mapping, and blueberry water stress. And for those of you who have seen this talk, the parts of this talk before, it's really broad brush, so you're not going to learn a bunch new about these things, but this is just to tell those folks who have not known this, that ecology has been operating in those 4 different areas, and I want to show a little bit of output from them. So this project has been focused on separating rockweed in the intertidal. Here's an image in the color infrared of the intertidal with some calibration panels here and some spectra taken from these little tiles here, showing what the different spectra look like. And this project, in collaboration and led by the Maine Maritime Academy, along with a collaborator at Bigelow Marine Sciences, as well as Schoodic Institute, are trying to quantify the biomass of rockweeds separated by species. And the remote sensing part of that project is led by a private sector company named Nearview out of Portland. And I'm going to get to private sector companies at the very end, because we don't have enough of them doing this work. But they're the one remote sensing team that we've been working with who does not have the sensors that we do, but has the capacity, is the Nearview. So we're working in collaboration with them, providing them both the expertise and the instrumentation to be able to collect spectra like this. But that will come back later. So anyway, the point here is that there's really different spectra of the different targets of the marine resources in the intertidal here. And I think there's a lot of potential here for us to measure new things like physiological drying as it responds to the drying every day, and understanding things like where the ecosystems are in these intertidal areas a lot better. There's a lot of organisms that survive and live in these places, and so quantifying the habitat is extremely important. And for the state of Maine, these resources are often, I guess, very litigated in terms of knowing who has the right to harvest there. And so it's important to protect them because they're so limited. And I know that the Wheatland lab is doing work in the intertidal as well. Next application, tree species mapping in the state of Maine. So obviously forestry is a huge part of both the ecosystem and the economy of Maine. And it doesn't, I don't need to go into why that's important. So then what you're looking at here are some spectral signatures of trees by species, a scan line from our drone, and then a subset of an image like this showing the same place in different wavelengths with little dots of known tree locations underneath them. And what this slide demonstrates is that we are working towards the tree species mapping and counting capacity, at least for the canopy. And the reason this might be important is, basic things like how many trees are there and which species are there are actually not that easy or even possible to answer at big scales. And so the tools that I'm talking about today are definitely some of the ones you would want to consider if your goal was to answer the broad question. I want to know every canopy tree species in Maine, the location and identity. And so in an image like this, the pixels can be assigned to individual species, but the problem is attributing the canopy to individual trees themselves. And so we're working on segmentation to take images like this, process them to a prediction, which we can do to almost 90% accuracy, and at least internally, and produce a map of not only which pixel is a tree belong to, but aggregate the pixels together into a single tree so that we can move towards answering questions like counting trees. Obviously, things like LiDAR would be natural to couple with this, and we have done some experimentation with that, but we're focused on the top of the canopy to be able to observe and segment and separate and predict what are these trees in this picture. And so again, the basic things LecoSpec does, what is there and how much of it, this is in the category of what is there. And the category of how much of something is there is another tree application that my graduate student, or actually Dan and I's graduate student, we're working on publishing this. It's getting close, but she graduated and published this in her thesis, which is showing ash health as a function of the electromagnetic spectrum. And healthier trees are in the lower number class, and sicker trees are in the higher number class. And these are scored on the ground with a professional forest pathologist. And we basically predict this health class from the spectra and then point that back at the map. And what you can see here is redder colors are supposed to be sicker trees, whereas yellower colors are healthier parts of the canopy. And then these other colors should be other things. And so about 70-something% of the accuracy, or the accuracy of this algorithm using these visible near infrared bands can explain about 70% of the variation in the training data that we used. And so we're optimistic we can use this tool to help answer questions about not just ash health, but also take more nuanced physiological measurements, which we're doing this summer using a LICOR to measure tree physiology with these instruments, and then possibly predict tree health in a more nuanced way, potentially by species. You would obviously be able to combine a species level map like I showed or indicated up above in the previous slide with something like this to be able to predict plant health in a very useful way. The next application and the last one is recently published in the Remote Sensing Journal. Catherine Chan, again, lead author there, graduate student, was able to show that in down East Maine, in a couple of fields next to each other that had been irrigated and not, you could really clearly tell the difference in the imagery and classify pixels very accurately based on whether they were in an irrigated area or not. But not only that, using a LICOR, we were able to measure, along with John Zhang's lab there at UMaine, leaf water potential. And then based on where those leaf water potential measurements were taken, use the reflectance of those pixels to predict the water potential elsewhere in the image. And so you can see that the orange area here, which is the unirrigated, has a lot lower water potential. Lower water potential. They're not sucking on the ground anywhere as hard as those that are unirrigated. So the point is that the separation of irrigated versus not is not good enough, separating the water stress and a continuous measurement is far more useful. And we were fortunate to have a collaborator in Wyman's, Bruce Hall, who was able to help us, locate these sites. And so that was a very nice use of these instruments. And so, just kind of to remind you that each one of these applications have different spatial scales and different spatial resolutions. And we have to remember that tiny pixels sometimes are needed, like the ones that I showed you, but maybe airborne pixels from like a plane or even satellite-based pixels would be enough. And, or we need those other pixels, these bigger pixels to scale. And I'm gonna talk about that next. So one of the ways to scale up from the imagery that we collected is to go from the drone up to an aircraft image that is collected over the top of the drone image. And some publicly available airborne hyperspectral platforms that you can download data from yourself include Avarice Next Gen, which is a visible infrared imaging spectrometer next gen. And there's also another sensor called G-Light at Goddard, which is another NASA center. And NSF operates the Airborne Observatory, which is basically the same sensor as Avarice. And this sensor is very similar to our sensor on the ground. It has much wider spectral bandpass, but the point is that it has also very big pixels. And so if you compare Avarice to our drone-based imagery, and try to like predict stuff, here's a quick snapshot on what that looks like. And this demonstrates kind of what I'm getting at with this trade-off with this spatial resolution. Here's 3 strips of Avarice imagery. Unfortunately, this is not from Maine, but there are forests and rivers and places that would be analogous. And what you can see here is these little tiles here are zoomed in areas, and these little tiles are UAV imagery from the same place that we flew, not far in time away from when the Avarice images were collected. And then we tried to predict the presence of a particular shrub that's important for moose in this case. And the white pixels are high probability that the shrub would be there. And so same in the UAV imagery. And so what I'd point out to you is things like this little string of what are willows in the red, green, and blue version of the same imagery. Here's that string of trees. Here's that string of trees. And for big objects like big swaths of willows, 4-meter pixels are fine. But when you have a smattering of willows or shrubs like this, the same location is quite different looking and doesn't really tell you the same type of information about the spatial relationships of the objects in present. And that's simply because, shrubs and trees can be smaller than 4 meters. And while that's not, shocking, the point is the error that you get by saying, is the thing I care about present or not, is the pixels get bigger, becomes a problem. And that's really the trade-off, again, that I was describing. And so if we have images like the ones that I'm showing you in which we can validate these places where the pixels are too big with these finer resolution images, we may be able to correct and scale up to these bigger images with our underlying UAV data. Now, of course, airplanes cannot fly everywhere, and there are now space-based versions of these same kinds of instruments. And the DLR, Earth Sensing Imaging Spectrometer, or DESIS, is on a module called MUSES on the space station, which can be pointed at things. And I have been spending my fall telling it to point at things in Maine. And I haven't analyzed any of those data yet, but they look kind of like this. So here's a tile I grabbed off of DESIS of a very familiar place, Mount Desert Island. And just for reference, here's some G-Light imagery, which is similar to Avarice, different in other ways. I won't go into it. But the point is that imagery from Maine is available both airborne and space-based. And so the kinds of scaling exercises that I think a lot of us are thinking about are available now for us to do. And the different band passes here just to sort of visualize things outside the color infrared. Otherwise, we'd be looking at, this kind of thing can be done with Landsat. This kind of thing cannot. And so the point here is that these data are available. And I think for the state of Maine to have more capacity to map stuff across the big areas that we live in, we need to start thinking with these other tools. And I know that some of us are, but it's a challenge. So this is one more slide borrowed from David Thompson. I want to point that one out again, that DESIS is on the space station. And for those of you who know about the space station, it doesn't go any further north than 55 degrees, nor any further south than 55 degrees. So polar regions are excluded. So other missions to be launched later, like SPG, along with these others from other space agencies, will provide coverage up into higher, longer wavelengths, like visible near infrared, but also provide global coverage. So right now we don't have global coverage and we only have visible near infrared. DESIS does not seek the shortwave infrared, so it only goes to 1,000 nanometers. So the future of these technologies for the state of Maine is bright, for the whole Earth for that matter. And so we need to be thinking about using these tools for our purposes. Towards that end, there are private sector solutions. Here's one, and it's not many, honestly, that operate imaging spectrometers on drones for the type of applications that LecoSpec does. This company is based in Indiana, and they're a startup based out of the sensor manufacturer that our sensor is from, Headwall. And so they partnered and created this company. But there's no reason we couldn't do the same thing here in Maine, and I think we should. There's also space-based versions of private sector imaging spectroscopy resources coming online. Peter Thiel, if you know who he is, he's a pretty controversial investor, but he just invested last couple of weeks in this startup called Hyperspeciq IQ, which is going to be doing, probably two satellites, it sounds like, for both military and civilian applications. We have not necessarily the exact tools to do that, but we have expertise in the state of Maine with a vegetable-based fuel rocket launching capacity, with the what's it called, Blue Shift. Wouldn't it be cool if Maine was thinking about doing its own kind of experimentation with these types of things? So, in conclusion, analytically, where I'd like to see things go with these technologies is to have more object-based stuff where the pixels are not only just being predicted to be things, but they're also being aggregated into targets, meaning like this tree, that tree, scaling up to the other tools like Avarice and G-Lite. Fusing these data with LiDAR would really be nice, and it actually is possible quite easily with sensors from the same company, Headwall. They sell a co-aligned visible near infrared, shortwave infrared, and 14-channel LiDAR system. So, it doesn't necessarily work for everybody, but, combining these technologies is where both the future is to answer questions, as well as I think, NASA is going that direction with things like SPG, which is going to be part of what they will call the Earth System Observatory, which will integrate sensors across different platforms like this. And of course, doing lab and field-based studies on diverse targets using imaging spectroscopy is something that I do and would like to continue. And I do a lot of work with folks at the University of Maine and would like to continue to do so, and think that these kinds of shared talks are places where we can do brainstorming, which leads me to my very last sort of set of content, which is, where do we need to go to do remote sensing of these diverse targets in Maine? Water and lands in Maine have many additional applications of these technologies. I've talked with people like Ivan Fernandez about doing things like soil health mapping. We've done some flying after snowmelt in fields to look at how much erosion and things is happening in large areas using drones, and why don't we do that with larger areas and think about using those spectra to estimate soil carbon and things like that. Marine and freshwater monitoring for algal blooms. I know folks are tasking DSIS to do this stuff. I'm not in the loop. And I know there's folks at UMaine who are involved with that sort of thing. We need new capacity, though, in our universities to develop these skills for these technologies. We are, it is racing ahead right now with these instruments and the machine learning and AI that's being developed to process their data. And I think we need to do a better job at keeping up. And to do that, I think we need public-private partnerships to incubate R&D into private businesses, or at least some sort of service that would allow people to use these data for Maine. And to do that, I think we need some venture capital and startup funds. And I know MTI exists, but I think there probably is a greater need. Peter Thiel put in 20 million to that one business. I think we need something on that order to do new businesses in Maine and New England to provide these services. And with that, I would like to thank you for the invite and let you ask any questions.
Thank you, Peter. Questions, anyone?
Well, okay.
Who's got
Thanks.
questions?
Go ahead, Doodoo.
You know, that Peter, that was a good pitch for the, public-private partnerships. So can you tell us a little bit more about kind of like your vision of how that would work in the market and stuff like that?
So what I like to tell people is if I were to get hit by a bus tomorrow, I'd want people to know that the idea is that, what we're doing, what I just showed you is on a shoestring, right? Imagine if there was even moderate investment. To be honest, we're coming at this from like a biologist point of view and realize the capacity here. So what I envision is just like what my students were able to do, one or two people with a truck and all these instruments can go and image hundreds and hundreds, maybe even thousands of acres, well, hundreds of acres a day for sure, with the drone-based stuff. So that's what I see. I see the paired instruments where they're going out and collecting spectra, they're taking samples, and they're sending them to the Maine lab to get analyzed. And there are many of these, as many that the market could support. And I understand your question, so let me answer it as best I can. So the market research that we've done, which is very minimal, but it is not zero, has indicated that pricing per acre is within the realm of reasonable compared to other things that are out there. The challenge I think will be going from the R&D to just building the clientele, honestly, and but I think I've given this talk, that kind of talk, to farmers. I've flown instruments for farmers. I didn't talk about some of the other things we've done just because the data weren't really ready, like potatoes. Many other crops, are very amenable to these analyses. And the other thing I didn't talk about is all the lab-based stuff that would give us the capacity to do things like at least teach people to work with the medical imaging world. People like Terry Yu in computer science came from that world. We don't have any training for that sort of thing. So the data, I think, is really the central theme. And I don't know if you noticed that on the private sector slide, but it said, we're not a drone company, we're like a data company. And I think that's what this company would also be. Drones are the flavor of the month, but where the data comes from is going to change, but the type of data is going to be pretty similar. We're going to have more and more of these imaging spectroscopy data sets. They're very large, they're complicated to work with, but if we were to work towards training students, and I think having then a pool of students would be available to then populate said business, I think the kinks would maybe be worked out by then. But I think that's part of the discussion I'm interested in having, is how do we get from where we're at now to a place where we have the surface that a researcher or an agricultural person or a farmer or a city could say, hey, you know what, we got a problem over here or a thing over here that it's of enough concern that we want to bring in these extra tools. Not unlike when you go and you get an additional, you get an MRI or get some additional scan. So I think that's really all it's not a complicated thing. I think it's just making the pieces work where the price is offered at a competitive price for what people need, as well as making sure that the outcome is going to be stable. And so we're still on the crux of like stable predictions for the things that we offer, like this tree, that tree, but there's no reason at the same time to be saying, hey, look, these technologies, they're legitimate. People are publishing them all over the place. And we could be subject to somebody else's business in the future. Rather than do that, why don't we build something from the ground here in Maine that we know works for us? So I think paying someone from out of state who has fancy instruments to come here now and then is not the model, because I think there's enough applications here. And I think, you know, within the Wheatland lab, I think there's space, but I think it's also that the Wheatland lab and everybody else is we're all under resourced. I think it's a matter of like bigger investment. I think that's the important distinction. The Wheatland lab does a great job promulgating geospatial information and doing these kinds of things, but nobody has $250,000 sitting in their pocket to go buy the instrument. You know, I think the Wheatland lab is why I'm giving this talk here, you know, is like they're the leaders. And I think, you know, it is kind of a money problem. I think that, you know, the future system, you know, and we've all been on those proposals with, we want to submit to NSF to get that money. But I think at the end of the day, it might be better to do both that and get some, convince somebody like a Peter Thiel in Maine to say, hey, look, you know, this is worth my money. And sorry if I'm misrepresenting anybody here, but yeah, I'm just thinking out loud that you know, the reason this isn't happening is because it's expensive and complicated, not just complicated. Thanks, and that was a long answer. Any other questions?
Peter, if I could maybe just briefly add to some of the complexity that you just spoke about. Personally, and I'm certainly not a hyperspectral expert like yourself, but I like to make the analogy of, you know, LiDAR technology, airborne LiDAR technology specifically, maybe 20, 25 years ago. You know, LiDAR machines have been around for quite a long while, but until there was a real convergence of technology and the integration of that technology to the point where it was robust, repeatable, it just worked, you know, all the time, every time, under many conditions, then it just exploded. And then your cost, you know, per unit area comes down to something that is really attractive to a wider audience. And I wonder if you know, maybe we're still on the cusp of that with hyperspectral remote sensing from a practical applications perspective. I don't know if you have any thoughts on that.
Yeah, I think the miniaturization of these sensors has rapidly, you know, expanded the capacity for people to use them, but they're still really expensive. And I think, you know, the mixture is what's really expensive. I think the really prohibitive cost is, and so if I were to add one specific thing that we could do in Maine, and we've done this in conversation with Alex Avarice in engineering, is build our own platforms. I think part of what's challenging is taking the spectrometers and putting them on things. And I think if there was a weak link at UMaine, I think that would shift the need maybe away from the geospatial scientists and more on the partnerships with some engineers. Because if we were buying spectrometers as a lab from the manufacturers and configuring them ourselves with our own, you know, GPS IMUs or just broadly kind of doing our more engineering in-house, I think we would maybe have more capacity. So maybe as a recommendation to leadership is to enhance, you know, our optics offering. Somebody like Ali, you know, who sits there at the nexus of those types of things. I don't know if he's on this, but that would be, you know, a great contribution, so that we would like, none of us are going to build those things. It'd be nice to have like somebody we could go to and say, hey, I want to build a version of G-Light that's cheaper, but put it on the Cessna, or I want to, you know, build a spectrometer for my lab. You know, I don't know where that sweet spot is, but I do think that we're there, and we can be, you know, we are. I mean, Doodoo and I and all of us, many of us in this call have worked on these data together and helped, you know, support the students to collect the data I just showed you. So I guess I shouldn't be saying we aren't doing it. I guess I want to say we should be doing more, and I hope that I convince some of you that it's worth it. That the things that you can detect and see, even if it's just sort of a glance, the potential is so great that it would be, you know, it's just a great set of tools. And then, of course, like the part about our jobs, you know, in the sector of both science and education is that we want capacity, and we don't have that. And so I think it's kind of a two-pronged thing. Like, hey, look how cool these are. And also we need capacity to grow this. And so, if you are interested in a public-private partnership, please do reach out to me. I think what this turns into is what we make it. It isn't just me doing these things. It's all of us. Many of the people on this call, or at least a lot of them, have interacted some way, shape, or form with these. So, yeah, I think it's a reflective question that I have. What do we want it to look like? Maybe somebody else will take what from this talk that they're going to write a grant and get their own stuff and start doing something. Or maybe we're going to come up with a way to package our ideas and go to the right organizations to say, hey, look, something different and more of a mixture. I think that maybe is a better approach, because just trying to buy the instruments and do it ourselves, whatever it is increasingly hard. We're all busy and so forth. So I think that's part of the reason why I think there's a space for a public-private partnership. Not to go on too much about the business side, but I think that that's important. I don't know if it's business. I think it's more that we need to get, if it was a different technology and people's lives were on the line, we would be saying, we need this technology in our hospitals today. So it's not quite that urgent, but it's similar.
Thank you, Peter. Any other questions for Peter?
Yeah, I'll jump in. I'll take it away from the that side of things and get back to the science. I like your talk, Peter. So I don't deal with any of this stuff, so I'm really kind of curious. And the species identification that you were talking about with those spectral signatures, I was just kind of wondering, how much do we? I mean, the way I always envision this kind of stuff is like you get a blast of information. You just you have a computer try to match it up the best you can. But how much do we actually understand about how the structure of the leaves or the chemical composition of the leaves really drives a fundamental that species identification? Have we gotten below the species level, where then for a subset of species you have these structural and chemical characteristics that you can then apply to other species, or maybe them, or is it just kind of a black box, squiggly lines, which one's line?
I think that's a doodoo question. I think that it's really, they come at it from traits agnostic of species, and use photochemistry directly, not like, oh, it's species A, so apply a different model. I mean, there may be a parameter for species in the model, but spatially, when we're making those kinds of estimates for our algorithm, it's definitely not based on the physics of how light will bounce around in something in a mechanistic model. We're using a correlative approach, but the way that we do it does give you a direction to look for where in the electromagnetic spectrum the answer is coming from. Doodoo, would you like to take that question though? I think you know that field better if you have time.
I mean, I was just going to say that, I mean, point blank stating that this is a doodoo question doesn't sound very good. But
well, I can tell you that you are a better expert at this topic, but I can give it a stab, which is that the traits are related more to, they're applied in a way that is not using species. That I know. And the way that we've related traits is not related to species. So, but with that, maybe you could give a slightly more nuanced answer.
So I mean, there are ways of diving into more the mechanism of, why can you detect what mechanistically or what physiologically and trait wise is different. And, we can do some of that. It's just, it takes a while and it's, complicated. You're doing either that with, physics, inverting a radiative transfer model, or doing, a lot of HPLC and chemical analysis, anatomy to figure that kind of stuff out. So it's certainly possible. And it's kind of like a cool avenue that's, I mean, it's not super explored.
I think the problem is that the applications that I mentioned were very specific and non-mechanistic. And so we were tuning something to a very specific application for a specific purpose. But I think there's still tons of basic science to do there. And, I don't think there isn't a good answer to that question, because there's so many different types of plants. I'll give one basic example and then stop talking, which is that in nitrogen, which is something we all know is important. We've heard people like Scott Ollinger lead us in that world for a long time, saying nitrogen in canopies of trees is related to near infrared reflectance, but we don't know why. As the pixels get smaller, do you get a better answer or a breakdown of where that connection is? And the answer, even just for that one chemical, since nitrogen is in so many different things, what would your expectation even be to have a homogeneous spectral signature of something that just happens to be in every protein? So I think we're really at a place in which it's a wide world of response variable. Like I said, information and light. I mean, you could just pick a different response variable and the chance of it needing somebody to study it is high. And that's part of the reason why there's urgency here, both from like the potential to answer problems, but also publish cool papers. I mean, it's a hot topic. So, but anyway, science-wise, I think there isn't a clear, like one size fits all. And you need to say, what is the question that you want to ask? And is there an algorithm that exists? If so, sure, use it. One of the things I didn't mention was that we could scrape intellectual property of the public sector to try to build this public-private partnership, just to have all these algorithms and pay undergraduates to code them up. And we could use all these different things that are out there, but that isn't happening either. So there's a huge potential there. And I think if your application is if I know we've talked about it, Jay, in the past, and you've come up in my mind many times, I would love to work with you on something to try to do kind of like what I'm talking about for other tree species. We're doing that with poplar up north with Ned Rupert Nason. We did some flying for him this summer. And so I think it's more just like, how do we, again, make this a collective who can operate these instruments? I didn't actually say that, but a collective who can operate these instruments in a way that everybody gets the data that they want. And I think that's kind of, you're a case in point. If you had a question, you got to work with me, and nobody wants to do with that. So why don't you, how do we keep people from having to work with me to get these data? That's really the goal here. Anybody else have any questions? We're 4 minutes over, but I can answer another one or two if there is one. This is great. I appreciate the interest.
I wanted to get your thoughts, Peter. I caught some of the flavor of it, but I wanted to see if you could expand a little bit more about now knowing you. Well, I don't know exactly what you do in your new job, but I suspect that that it involves a little bit more interaction with the general public and citizen science and that kind of thing. So I'm wondering if you had any thoughts about how you translate all of this complicated stuff that you were just talking about into that environment.
Well, I actually don't have to do as much citizen science stuff as you thought you'd think, but I am doing citizen science with this stuff. And I think the way that I think about it in that domain is like, what's in that pixel, right? Is it a big pixel? Is it a small pixel? And if you have more colors in that pixel, it tells you what's in that pixel. And I do see and have worked on proposals that may be funded about taking that logic and applying it to validating pixels so that, once we get these space-based things and we have all this data, what are we going to do with it if we don't know what's in the pixels? That's a huge endeavor. And once these space-based things are everywhere, it's just going to be a big endeavor to make sense of the pixels. It's the same thing that we were just kind of addressing with Jay. It's like, what is the information I'm getting out of this? But anyway, the point is, like, the citizen part, I haven't. I'm actually trying to be more of a science liaison at Spacebased and sit at the level that I'm at right now at this conversation, and less involved with citizens because this has been my choice. But we're actually moving into more use of DESIS to do monitoring for the Park Service. That's the direction we're going, as well as using the ground validation data sets that I've mentioned that I'm trying to build and make public, like spectral libraries of targets. And I didn't even mention that, but that's actually a huge missing piece. And so that's where the citizens can come in, I think. And so as I get involved with citizens, that's kind of the direction I'd like to go with this particular set of technologies, because I think there's a ton of potential, and the communication potential is really high too. Like, everyone can relate to light. And I think that's something I tried to do in this talk, is just use simple words. So I don't know, I'm trying to work on that question. I don't have a good answer, but that's where I'm at with it now. Anybody else? Any other questions? I really appreciate it, all the interest. I was really sweating this talk too, by the way. My kid's been sick the last 3 days, so. I hope you enjoyed it. Tawanda, I see is on here. I don't know if you have any questions, or you can tell me I'm totally wrong about half the things I said. I'm sure that's what. Anybody?
Well, thank you, Peter. That was wonderful.
Oh, Tawanda, were you going to say something?
Oh, no, I just wanted to say that it was a good talk. And yeah, I mean, I saw that we have got a lot of areas that we can share, especially, I mean, because that's also my area on hyperspectral remote sensing and trades. So I think it was a good talk. And thank you for the talk.
Sure. Yeah.
Thank you so much. All right.
Thanks for attending. And yeah, Tawanda, I think, has a lot, he's really he's an example, actually, when I met him, of like what Maine needs to do. They need to hire people like Tawanda as scientists to help make, we need more of us. So I think maybe it's happening. People like Pari, hired him, and we need more of him and more people to do the work. But we also need technology. And the last thing I'm going to say, and maybe this can be a good parting call, is like, there is a group of Maine hyperspectral people. It's de facto, they're in this meeting, and maybe it's time that we make that something that is a stable thing, in which it's separate because it's different enough, the words are different, all the things are different. So I would put out there, if there's interest, maybe we can have a self-organized thing like that happen at some point. Anyway, thank you again for the invitation, everyone. Yeah, it was really a pleasure.
Great. Thanks very much, Peter. Thanks everyone for joining us. Have a good afternoon.